Bill. Oh, yeah. Okay. But it's interesting. One thing I'm more encouraged about ticks is whether or not we have a testing site here. I actually said, when I had
That's really good.
Just be sure if you're using the bathroom, use one that the first one.
I can't really take them out of Santa Cruz, but this is. Yeah.
We'll get started in 2 minutes.
Great.
Yes, I can stand here for this. You can eat, yes. This is one of the weird kind of hybrid rooms. The rule when I was at lab tech here was you can eat as long as there's not like chemicals and fish and other things that are out.
That's fine.
Yeah, I'm not 100% sure what the kiosk. I haven't kept track of the parking. Yeah, sorry. I don't know if it's necessarily this time of day or not, but. Oh, it is, okay. Yeah, that's one of the challenges of the parking. Let's wait till it's back. It's okay if I get started?
Yes.
Okay, great. Well, hello, everyone. Thank you so much for coming to tonight's talk. My name is Joshua Benish. I'm an associate director within the Office of the Vice President for Research at UVM. I support work around the Water Resources Institute and the UVM natural areas and mountain environmental science, and have been helping support this Talks and Walks on UVM's Wild Side lecture series. We have a lot of key partners with this talk, and if you haven't noticed, already had some materials out to the side. But all of our key partners are the UVM Natural History Museum, the UVM Natural Areas, the Rubenstein Research Forest, the Rubenstein Ecosystem Science Lab, the Field Naturalist Program, the Bringle Herbarium, the Consulting Archaeology Program, and the Environmental Program. And typically, we've been having these talks over at ECHO, but this month they're working on all of their aquariums. So we appreciate you coming here to the Rubenstein Lab and kind of experiencing what a UVM lab looks like. I worked here for 5 years as a research technician right across the hall, so I'm pretty familiar with the space. And so yeah, so thanks for joining us for this talk, another Talks and Walks on UVM's Wild Side lecture series. This month we welcome Dr. Ellen Martinsen. Dr. Martinsen is an adjunct assistant professor and lecturer in the Department of Biology. Her research interests center on host parasite ecology and evolution, wildlife disease, avian biology, and conservation. She manages the Wildlife Pathogens Lab at UVM and uses the UVM Centennial Woods natural area for some of her tick disease-related research. Today, she will be presenting to us about Vermont tick-borne pathogen ecology. Now I will hand it over to Dr. Martinsen. Thank you so much.
Hi, folks. Thanks for those online. Thanks for everybody here. Thanks, Josh, for inviting me. Thanks to the Office of the Vice President for Research for organizing, and then thanks to Echo for hosting. I've actually been in this lab cutting fish, looking for fish parasites last spring semester. So again, my name is Ellen Martinsen, and I run the Wildlife Pathogens Lab and the Department of Biology at University of Vermont. I just want to give a big shout out to my lab members. So we have a handful of undergraduates and 3 grad students in our group. We look at host parasite ecology, wildlife disease, mostly vector-borne pathogens transmitted by mosquitoes and ticks. And so that's who we are. There's our site if you're interested in learning about more. And so this is actually a team effort. So I'm giving the tick talk today, but actually this work is a collaborative effort of multiple, mostly undergraduate research students in my lab, as well as some grad students. So I've had amazing undergraduate research students take on small facets of this collaborative research as their senior research projects in the lab over the years. So starting with Sam Cranston, Anna Henkel, A.D. Pinnell, Olivia Bassetti, Joe Webb, Leah Carmen, 2 grad students in our department, Lauren Berkeley and Emily Beasley, who have graduated, have helped procure some of the small mammal samples that I'll be talking about today. And then finally, my collaborator and friend, Marm Kilpatrick at University of California, Santa Cruz, is a disease ecologist and ecological statistician, and he's helped make sense of the data. So funding, I'd like to start with funding. Again, this work has been primarily pushed through funding for undergraduate research. And so the UVM Office of Fellowships, Opportunities in Undergraduate Research, or UVM4, has provided those students on the previous slide with summer research fellowships and research funds to project this work forward. As well, we've also gotten grants from other places at UVM, including College of Arts and Sciences, College of Ag and Life Sciences, and our own Department of Biology. American Society of Mammalogists funded the grad student work. And then finally, the UVM Natural History Collection, I'll be talking about it in more detail later, but has provided us with countless intern hours, countless specimens that we've used, as well as supplies. And the UVM Natural History Collection is curated and directed by Dr. Sarah Helms-Cahan at the university. So just an overview. Today I'm going to be going through an introduction, then major research objectives, results, an overview, and then open it up for questions at the end. And likewise, we'll be monitoring questions online for those of you who are attending virtually. So, and it's needless to say that we're living in an age of emerging infectious diseases. This is a map put out by Anthony Fauci in 2017 of emerging and re-emerging infectious diseases in humans worldwide. Deliberately emerging down at the bottom, that is just one pathogen, and that's anthrax. But all the others are emerging or re-emerging in nature. Here's a picture of Anthony Fauci in 1984 when he started as director of National Institute of Allergy and Infectious Diseases. He served that role for 4 decades, and at the end he concluded, the emergence of new infections and the re-emergence of old ones are largely the result of human interactions with and encroachment on nature. So that's how we kind of left it after 4 decades working with human infectious diseases worldwide. And so what I like to emphasize is that the majority of these infectious diseases on this map are the result of pathogen spillover from animals. So these are zoonoses or zoonotic pathogens that are spill over into humans from animals. So scientists estimate that 3 out of 4 of every 4 new or emerging infectious diseases in people spill over from animals. So that's something to keep in mind, especially as I go through my talk today. If you haven't read the book Spillover by David Quammen, it's a little outdated, but he actually predicted our pandemic with SARS-CoV-2, COVID-19, and he spent many years traversing the globe talking with medical doctors, villagers, epidemiologists, you name it, getting to the bottom of the emergence of major infectious diseases in humans worldwide. It is one of the best reads. I have it as an audiobook in the car, and I could listen to it endlessly. Students in my lab are required to read this, and it literally changes the way that they look at the world. So if you have a chance to read Spillover, please do. So we're all pretty familiar with these pathogens that spill over into humans, these zoonoses. So we all know about malaria, avian influenza, It's now in the US, it's now in our dairy cow in the US. Lyme disease, Chagas disease, Ebola, SARS-CoV-2, COVID-19. That's a spillover from animals into humans. And then, fantastically, it spilled from humans back into animals. So zoo animals are getting it from infected zookeepers. And now white-tailed deer across the United States have SARS-CoV-2 viruses circulating among them, which spilled over from humans. And then more recently, Triple E has been in the news. We're probably all well aware of this one. This is a pathogen spillover. So Triple E is a pathogen that's native to birds. It's endemic here in Vermont. It's been here for a long time. It didn't just show up. It's transmitted between birds by a bird-biting mosquito, and it very rarely bridges by another vector into humans. We are accidental hosts as well as horses. And so this is a case of pathogen spillover. So if you've had any of these diseases, you've been the recipient of a spillover pathogen. I have to put a plug in here too. Emerging infectious diseases are raging worldwide, increasing in incidence in wildlife as well, to the point that they're causing population declines, population endangerment, and even species extinctions. So they're on everybody's mind. So we go back to that Fauci map, and I put new emerging or re-emerging diseases in the US since 2015. These are just some of them that we can add. So we are just seeing this onslaught of emerging infectious diseases. And if you look at these, the majority of these are actually vector-borne diseases. And so vector-borne diseases are emerging at a growing rate and are disproportionately causing new infectious diseases in humans. So I'll be talking about vector-borne diseases today. Why are vector-borne diseases on the rise? There are innumerable factors. Some of the main players include climate change, which are long-term shifts in weather conditions and patterns of extreme weather events. We are all familiar with these events in Vermont. Our flooding, our temperature yo-yos in the winter. For any skiers, we'll get a beautiful snowfall. The next day it'll be above freezing, then it'll flash freeze and rain, and then we just have crust. So this is all climate change. Land use or land cover change, as well as habitat fragmentation, are also factors that have led to the increase in vector-borne diseases. Can anybody guess which type of vector is responsible for 95% of human vector-borne disease cases in the US? Yeah. Mosquitoes. Good answer, but that's not correct. There's another type of vector in the US that's responsible for 95% of our human infectious disease, vector-borne diseases, ticks. So that's right, folks, ticks. 95% of vector-borne disease cases in the US, ticks. And what's happening with ticks in the US? They're increasing in abundance, they're increasing in range. So this is the Lone Star tick. Has anybody heard of the Lone Star tick? You can see it is expanding its range. Traditionally, it was distributed in yellow, and now the more dark yellow is where it's expanding its range. It's just shown up in Vermont. And this tick species transmits alpha-gal syndrome, which causes the red meat disease. So beware for Lone Star ticks. Another tick which has made its way northward over time in the US is the black-leg tick, also known as the deer tick. I'm going to just be referring to it as the black-leg tick for the remainder of my talk today. But you can see over time, over the last century, how it's expanded its range northward. So this is because of climate change, habitat fragmentation, and land use changes. So if we look at tick species in Vermont, this is our list. We have 15 species of ticks here in Vermont. 2 of them are new. So the Lone Star, a recent arrival, as well as deer ticks or black-leg ticks. We traditionally did not have this tick species here. So the history of black-leg ticks in Vermont, first documented in a deer in southern Vermont in 1985. Our state entomologist first field collected them in southern Vermont in 2004. First Lyme disease cases documented in 2005. And the last reporting of Lyme disease cases in the state for 2022, 1,300 folks in the state had Lyme disease. That's likely an underrepresentation of the true incident rates of Lyme disease in Vermont residents. And if we look at the distribution of Lyme disease cases, you can see they're all across the state. We have these ticks everywhere. So this fantastic creature, the black-leg tick, Ixodes scapularis, has 4 life stages in its life cycle. So this is an engorged female. Engorged female will spit out 1,500 to 2,000 ticks in one spot. She'll do that in the spring, they'll hatch in late summer. And they usually don't go more than 1 to 2 meters from where that egg mass is laid. And they are going out and host seeking usually small mammals and small birds. And so then they become this stage right here. I don't know if this is gonna work. No, okay. I don't know if there is a pointer, but this is the eggs hatch into larva. The larva takes a blood meal and then molts into the nymph. And then the nymph takes a blood meal and molts into the adult. This is adult female. This is an adult male. This life cycle takes 2 years in nature. So here is the life cycle. So from egg to adult, that's 2 years development of this tick. That's a long time for this vector to develop in nature. If everybody can hold up their index finger and imagine, this is the size of black-legged ticks. They are incredibly small. They're smaller than other tick species that we have here. And you can imagine it's these very small stages, the nymphs, that are responsible for most Lyme disease cases in humans. So if we look at Lyme disease cases in Vermonters over the months, you can see there's a spike of infections in the winter. In the summer, this corresponds to when the nymphs are out post-seeking. And so they're difficult to find, they're difficult to see. This is how most people get infectious diseases from ticks. And now, because of climate change, they're questing or host seeking year round. So you can't actually get away from ticks any year of the month. We've picked ticks off our dogs every month of the year. So if the temperature gets above 34 degrees Fahrenheit, you will have questing adult ticks in the winter. Horrible. So I want to talk about my history with black-leg ticks. This is how it happened. So my oldest daughter, 3 years old, in the forest next to our house, building a fairy house, amazing construction craftsmanship, I have to say. But the next day I was brushing her hair and I found a black-legged tick embedded behind her ear. And we had never seen ticks in our town before. So I wasn't doing tick checks. That was the first black-legged tick I ever saw. And if you've ever pulled an embedded tick off of yourself or a family member or a dog or a friend or a colleague, which I've done all of that, it's a horrible thing. Just to know that this vector is in unison with your bloodstream, with the bloodstream. So you don't know what's going on. And if you're wondering why it's so difficult to pull these buggers out, I took a picture under my light microscope of the feeding tube of a black-legged tick. You can see it's barbed and the barbs point upward. So if you're ever wondering why they're so difficult to get out, this is why. So pull them out very, very slowly. So traditionally, I was sending ticks that I pulled off my kids and dogs. I will admit I did that to tickreport.com. It's a tick testing company down at UMass Amherst. This is a tick that I pulled off of my dog. It was a fully engorged deer tick, and they'll test it for various pathogens and send me back the results in 2 or 3 days. It's great. And so you can see this tick actually tested positive for Borrelia as well as Anaplasma. So 2 different tick-borne pathogens that had a co-infection. So then my mind kind of blew, like, whoa, what's going on? This is a tick from our backyard. So then I had a very motivated undergraduates want to do a senior research project on ticks. He's from Martha's Vineyard, and everybody in his family had recurring Lyme disease. This is Sam Cranston. We went out, we started collecting ticks and screening them in the lab. And then lo and behold, my kids turned into tick collectors on their own. So when asked, do you want to stay home or do you want to go out tick collecting with mommy and students, they actually want to go out tick collecting. It's that much fun. If we look at the pathogens transmitted by black-leg ticks that cause diseases in humans, there's 8 of them. They're listed here. For the purpose of my talk today, for our research only focuses on 3 of these pathogens, Anaplasma phagocytophilum, Babesia micrate, and Borrelia burgdorferi. I'll talk about them individually later. But I want to remind you again, these diseases are spillover. So ticks hatch from eggs as larvae, and they are uninfected with these pathogens. Ticks are not the enemy here. They are born uninfected. They pick up these tick-borne pathogens from reservoir hosts in nature. So the animals sourcing the infection are actually small mammals and other mammals that the tick encounters on this 2-year life cycle venture. And so not only do they accidentally infect humans, but they're also pretty common infecting other domestic animals like dogs and horses. And there's so many variables at play in tick-borne ecology and tick-borne pathogen ecology that I've been to a conference and somebody called it stupidly complex. It's really actually stupidly complex. We have climate factors like rainfall, temperature, but even humidity in the leaf litter at the bottom of the forest. That's so important for these ticks. Vegetation, do we have full canopy cover, partial canopy cover, host density, habitat, host diversity, how many mammals do you have in your environment? And so it's really complex. And so you can see here then that the health of humans is tied into the health of the animals in the environment, which is tied into environmental variables. So this is when we lock together different disciplines under this umbrella, it's been coined One Health. So we really need a One Health approach when looking at tick-borne diseases. And even it's bust out of the One Health realm, and we have a new discipline called planetary health, because there are other factors at play here with tick-borne diseases in humans, like socioeconomic factors, development. There's so many things that this is really a planetary health issue. It's all encompassing. And fortunately, UVM has just launched a planetary health initiative. And so we have some awesome projects coming up with cross-disciplinary research across colleges and across departments. So it's an exciting time. So going into the three pathogens and the diseases they cause, Lyme disease is caused by Borrelia burgdorferi. It's a bacteria. Here's the number of Lyme disease cases in the Northeast from 2000 to 2017. You can just see it's been climbing north. There's a number of symptoms associated with Lyme disease. I probably don't need to go over these with you because we're all living in Vermont. Anaplasmosis is caused by Anaplasma phagocytophilum. That's another bacteria. And reported cases in Vermont have been growing exponentially. We have a gap for COVID years when we weren't reporting similar symptoms like fever, chills, headaches. And it's not just in Vermont that these pathogens are increasing in incidence. So anaplasmosis has been surging in humans up and down the eastern seaboard. So in Maine, and then the graph to the right, where it's growing, that number of infected human cases is going like that, that's Pennsylvania. So this is definitely a regional problem. And I want to remind you too that these pathogens don't just infect humans. So anaplasmosis is actually a really common disease in dogs. And so the upright picture is a recent graduate from my lab. Anna took this picture. She works in a vet clinic here in Chittenden County and sees infected dogs every week with anaplasmosis. I couldn't help put in a picture of our pack of dogs here, all rescues from the South. We haven't had anaplasmosis yet, but we've had Lyme disease in them. And then horses are really prone to anaplasmosis. And there's actually great data from dogs. So these are graphs provided by the Companion Animal Parasite Council. And they are test results from routine blood work that's done when you bring your dog to the vet every year. So in this left chart, you can see in 2012, in the state of Vermont, 1 in 3 dogs tested positive for anaplasmosis. Fast forward to 2024, 1 in 3 dogs test positive for anaplasmosis. So if we're looking at the surge in anaplasmosis, it's not just humans. Again, this is a One Health issue. Babesiosis, this might be my favorite one. So it's caused by Babesia microti. This is a single-celled protozoan parasite. This is a single-celled animal. This is a parasite that makes its living off of parasitizing hosts. It lives in the red blood cells, and you can see the number of cases have been increasing in Vermont. But as with anything in biology, ecology, we can't think about anything in unison. Everything is connected. We are all interconnected. These pathogens are interconnected. And so there's been more focus on co-infections, because we can't expect these pathogens to exist in singly. So co-infections are a poorly understood phenomenon in the tick-borne disease realm, and they present complications in clinical and laboratory diagnosis, and they likely increase disease severity. So it's estimated, it's documented that 4 to 5% of patients with Lyme disease are co-infected with either anaplasmosis or babesiosis in Lyme endemic regions. That's Vermont, that's the eastern seaboard, that's the Midwest. So these things are not occurring by themselves. Co-infections are actually much more common than we previously knew. And this is a news, just a study came out this summer detecting co-infection with two tick-borne pathogens for the first time. So the more that we look, the more we find. This is a list of, that table of symptoms in these patients that had these co-infections, it's a very long list of symptoms. So you can imagine how difficult it would be to be a medical professional and be diagnosing this disease. And so these clinical conundrums, it's not just here in Vermont. So these 3 tick-borne pathogens and these diseases are actually very common in Europe and other areas in the US. So it's not just here that we're pressed with the emergence of tick-borne pathogens. And in research, folks have been finding that these pathogens actually may not be randomly assorting or randomly associating in tick hosts. They may be found together more often than not if by chance. And so this is something I'll be touching upon today. Reservoir hosts. Again, these ticks have this 2-year life cycle, and they're taking a blood meal after they hatch out of the egg as larvae. They don't go far from the egg mass. That's their first blood meal, and then they molt into the nymph. And these are the critters in the summer that are most responsible for Lyme disease cases. The nymph has taken 1 blood meal. It sucked the blood of 1 animal. Whereas if you get bit by an adult tick, it has taken a meal as a larva and a nymph, and so it's had 2 blood meals. And so what do we know about the wildlife species in nature that are sourcing the infections in ticks? We know that white-footed mice, Parameciscus leucopus, are the most competent reservoir hosts. They're kind of indicated as the main reservoir sourcing these infections, but less work has been done on other small mammal species in nature. What do we know about tick-borne pathogens in Vermont? I know that everybody's eyes will get wide here. So we have statewide surveillance every summer by Vermont Agency of Agriculture, Food and Markets. The towns that they study every year change. For 2023, they sampled the towns shown here in green. And here are the tick testing results from last summer from 2023. They screened a total of 205 ticks across the entire state of Vermont. That was 58 nymphs and 147 adults, all blackleg ticks. 33% were found infected with Borrelia burgdorferi, that's the agent of Lyme disease, 7% with anaplasma, 4% with babesia. And then if you look at co-infections, they did find some co-infections. But if you look into the data, into the towns, they only actually sampled 7 ticks total from Chittenden County. So here we are in Chittenden County. This is the most populated county in our entire state, and we have data from 7 ticks. So we're definitely lacking data on tick-borne pathogen prevalence here. So that leads into my major research objectives. 1, investigate the prevalence of these 3 tick-borne pathogens in blackleg ticks in Chittenden County. 2, investigate co-infections in Vermont blackleg ticks, including possible non-random associations. And then 3, identify potential reservoir hosts for the pathogens, including co-infections. So these are our sampling sites. There's 7 of them. And our goal was to represent a gradient from urbanization to Green Mountain National Forest. And so we sampled sites you can see here. We sampled all tick life history stages. And a big kudos to the sites and landowners that allowed us to sample. So UVM natural areas, St. Michael's College, Green Mountain Audubon Center. And we have amazing neighbors that let us go through their property collecting ticks there, as well as some farmers in Sheldon. So how do you collect ticks? It's super simple. This is where my 7th grade home economics sewing skills come out. I still have my sewing machine from 7th grade, and I sew these tick cloths because you can't buy them online anymore. So it's just 1 meter by 1 meter flannel cloth. I sew in some weights in the bottom, put it on a wooden dowel, and you drag it through tick country. That is tick country. That just screams stay out to me. We put them in ethanol and we bring them back into the lab at UVM. So UVM's own Centennial Woods Natural Area is one of our field sites. And this is an amazing resource, not just for the UVM community, but for the greater Burlington community. I can't tell you how many people we have seen when tick collecting in Centennial Woods. Not only do you have hordes of UVM students out there doing labs on bird behavior or tree diversity, but you have students just recreating, just reading a book out there. I've seen preschool kid groups in there. I've seen running groups in there. There's just a diversity of people that use this property. It's such an amazing resource right in the middle of Burlington. So here's our lab group about to collect ticks at Centennial Woods. Another one of our field sites is Jericho Research Forest, another property owned by UVM. And so we went out collecting ticks. These are the undergrads who helped tick collect. And I just want you all to look at the smile on the student on the left, Sam Cranston with the blue baseball cap. He there collected his first nymph, and can you see the joy on his face? It's very satisfying to collect these things. And so we were able to sample and field collect 209 nymphs and 321 adults, so a total of 530 ticks. For sampling potential reservoir hosts, I relied on samples provided from grad students. So this is Emily Beasley. She graduated with a PhD from the Department of Biology. She had a study on white-footed mice, and she was already collecting tissue samples, ear clip samples. You can see the deer ticks attached to the ear of this deer mouse. This species is recognized as the most competent and important reservoir host species in the Lyme disease realm. So numerous Lyme disease studies have shown that in areas where you have dense white-footed mice populations, you have increased prevalence of Lyme bacteria, as well as anaplasmosis and babesia in the ticks, as well as an increase in the incidence of infected humans in that area. So this is a very important host species. To get at other host species, we utilized the collections at the UVM Natural History Museum, specifically the Zadok Thompson Zoological Collections, which are hidden gem. Did anybody know that UVM has a natural history museum? Unfortunately, we don't have a space to have exhibits to the public. Fingers crossed, maybe in the future, that'll be something that'd be amazing. It houses over 130,000 insects, over 8,000 mammals, and over 2,000 birds collected from a broad geographic span, as well as an immense time span. So it's an amazing resource. We have intern courses in there, and we also receive bycatch from some ecological studies in Vermont. So we're getting small mammals. This work is in collaboration with the Vermont Reptile and Amphibian Atlas, as well as Vermont Family Forests. And this work was led up by Lauren Berkeley, who just graduated with her master's, and a number of undergrads who helped sample tissues from small mammals. And through the museum, we were able to access 150 tissue species from 9 small mammal species in Chittenden County Forest. So these are not white-footed mice. These are all the other small things that scurry through the forest, and I'll talk about them more in detail later. In the lab, we bring our mammal sample or our ticks. We perform DNA extraction. We do a multiplex PCR, and that basically tells us if we have the presence of DNA for the 3 pathogens. Sometimes we have the presence of all 3 pathogens in a sample. We found a number of triple infections. And so results on prevalence. So here on the top, I show prevalence. That's percentage of infected ticks. On the top are adults, and the bottom are nymphs. You can see we didn't sample adult ticks and nymph ticks at all sites. And the pathogens, Borrelia burgdorferi, that's the agent of Lyme disease, that's the blue bar. That's the really tall bar, high prevalence across the board. Anaplasma phagocytophilum, that's in green, that's in purple, and then Babesia microti, that's in green. So you can see that there's significant differences between Borrelia and Anaplasma and Babesia, and actually Anaplasma is significantly greater than Babesia. And interestingly, these pathogens didn't differ in prevalence between sites except for Babesia. Some of our sites, we didn't find any Babesia in the ticks that we sampled. So these are the data. For adults, 65% tested positive for Borrelia burgdorferi, that's agent of Lyme disease, 9% for Anaplasma, and 4% for Babesia. If we look at our nymphs, 33% of them have infection with Borrelia burgdorferi, 9% with Anaplasma, and 4% for Babesia. Compared to the state results, they found 33% of their ticks infected with Borrelia burgdorferi. We have almost double that in our adults. For their analysis, they coupled adults and nymphal ticks, but for us, we separated it out. So we're finding higher prevalence of Borrelia, but not so much for Anaplasma and Babesia. But something that's starkly surprising that we didn't expect to find is that we only found a significant difference in the prevalence between adults and nymphs for Borrelia burgdorferi, and not for Anaplasma phagocytophilum or Babesia microti. This is really surprising because if you remember the complex life cycle of the tick, the nymphs have only had 1 blood meal and the adults have had 2. So you would expect higher prevalence of these pathogens in the adults, but we didn't find this for Babesia and Anaplasma. We only found it for Borrelia burgdorferi. So this alludes to the nymphs picking up babesia and anaplasma in nature as larval ticks. So we then looked at co-infections and we used a pairwise comparison to determine expected rates of co-infection based on single infection percentages. Basically, we wanted to know, are these pathogens associating randomly, or are they found together more often than not expected by chance? So we found marginal evidence that babesia and Borrelia increase infection with each other in both nymphs and adults. So this is pretty confusing, but if you, the rate of infection in ticks is 40% with Borrelia if the tick is negative for babesia, but in the ticks positive for babesia, their prevalence with Borrelia is 57%, and it increased across the board. And so these data suggest that there's an association between Borrelia and babesia in our ticks. They're not randomly associated. They're found together more often than expected by chance. And likewise, we found stronger evidence for association between anaplasma and babesia infections in ticks. For this data set, because the prevalence for anaplasma and babesia didn't differ between adults and nymphs, we could group all the ticks together. We had greater statistical power, and we found a significant association between these two pathogen groups. So specifically, the fraction of ticks infected with anaplasma was 4% if the tick did not have babesia, but if the tick had babesia, then it was the prevalence or fraction of infected ticks was 20%. So your infection likelihood with the other pathogen increases if you have the other. So it may be that these pathogens are facilitating infection with other pathogens, or it may be that they're picking up these co-infections from nature. So again, mixed infections, we found them at all the sites sampled. We found them in nymphs and adults. We even found triple infections. So some of these ticks, nymphs and adults, had Borrelia, anaplasma, and babesia, which was pretty scary. So what the heck is going on? Then we were asked the question, who might the potential reservoir host be? So here is our data from the white-footed mice. Again, this is the defined competent main primary reservoir host for Lyme disease and the other pathogens. And we found a 55% Borrelia burgdorferi prevalence. So over half of the white-footed mice that we sampled from sites in Chittenden County are infected with Borrelia. So those little deer mice are highly infected. This is super interesting. But surprisingly, we did not find babesia or anaplasma infection in white-footed mice. So that begs the question, who the heck are these ticks getting their infections from? You go out into the woods, sure, there's going to be white-footed mice, but they're not picking up their babesia and anaplasma infections from white-footed mice. So these are the other reservoir hosts, other small mammal species that we sampled. There's voles and shrews and jumping mice and moles, all these little brown things scurrying around on the ground that we do not give much attention to. But if you find a pathogen listed under it, it means that we found that pathogen in that small mammal species. If that pathogen is bolded, it means that we found that pathogen in that mammal species for the first time ever. So we found a number of new pathogen associations with small mammals in Vermont small mammals. And you can see that some of them were highly infected. Woodland jumping mice, 60% had Borrelia, 7% had anaplasma, 24% of them were infected with babesia. And those in red boxes, these are small mammal species that had co-infections, which is interesting because primarily we thought that these critters, the white-footed mice, were sourcing these infections in our ticks. But these data and our results kind of shed new light on there's actually increasing complexity here, and there may be a number of other key players in the reservoir community that we've been overlooking, like shrews and moles and voles. Who knew? And traditionally, Lyme disease research projects set traps, they bait them with seed, and so we are not catching all of these small mammals. They're hard to catch in the traps that we use. And additionally, some of them are insectivores, and so they're not going to go to a trap baited with seed. So they're not commonly sampled for tick-borne pathogens. And again, these are the reservoir hosts for these pathogens that are spilling over into humans. So quick overview, we found higher than previously reported prevalence of Borrelia burgdorferi infections in blackleg chicks in Chittenden County. we found twice double the infection prevalence previously found by state of Vermont. Significantly greater prevalence of Borrelia burgdorferi in adult versus nymphal ticks. Black-leg ticks are likely picking up babesia and anaplasma infections as well as co-infections as larval ticks. So they hatch out of the eggs, and that is when they're probably getting these anaplasma and babesia infections. Babesia and Borrelia, as well as babesia and anaplasma, are non-randomly associated in ticks. We're finding them together more often than not. White-footed mice have a very high prevalence of Borrelia burgdorferi, so watch out for those deer mice. Other small mammals may be reservoir hosts for anaplasma and babesia, as well as co-infections. And so this points to a diversity of all their small mammal species, including these jumping mice, moles, voles, and shrews, are really important players in the tick-borne pathogen realm that we have been overlooking up until now. And so this is pretty important. So again, white-footed mice density is highest in disturbed areas. And so habitat factors and fragmentation and habitat change is playing a big part here. We know that these are the primary reservoir hosts for Borrelia burgdorferi, but we really need to start thinking about modification of environment and environmental cues in terms of how they shape the composition of other small mammal species in their environment. So there's a lot more work to be done. We need to be screening more mammal species, and we need to be doing more reservoir host surveys. This picture creeps me out because you can tell this is somebody walking in the woods in the fall. You would not find me walking in the woods in the fall like this. I don't touch the brush to the side because, again, what we found is adult ticks are going to be questing in October and November. They love it when the temperatures drop because they know they need to get that blood meal before the onset of the winter. They overwinter and lay their eggs in the spring. But in our forest, the forest that we sampled in our study, 65% of adult black-leg ticks harbored the pathogen Borrelia burgdorferi that causes Lyme disease. 9% harbored infection with anaplasma effect or cytophyllum, which causes anaplasmosis, and 4% have Babesia microti that causes babesiosis. That's not even talking about co-infection. So a lot of these ticks don't just have one of these pathogens, but they have multiple. So going forward in our lab, the wildlife pathogen lab, we're including more sites. In addition to what I presented today, we want to collect more ticks. We want to sample more mammals. So we're building more collaboration. We want to screen for more pathogens. I just included 3 today, but there's a whole suite of others, and we're discovering more and more pathogens in black-leg ticks every time we look. So we'll be doing more grant writing. Fortunately, I have a fantastic PhD student in the Department of Biology that I co-advise with Nicatelli, who's working on the project, Carlos Amisa, and here he is holding a white-footed mouse. And we've just bridged a collaboration with the New England Center of Excellence in Vector-Borne Diseases. This is a group down at UMass Amherst that's funded by CDC, and so they'll be helping us run some of our tick and small mammal screenings because we need more funding. And so there's promising tick. I don't want to leave you hanging in dismay about the problems these tick-borne pathogens. So there are some really promising disease projects, research that's come out. This is out of the UMass Amherst group, led by Steve and Rich. They do amazing stuff. So a big question is, why don't deer get Lyme disease? So I've been talking about black-leg ticks and deer ticks. They're called deer ticks because they reproduce on the host. So the male and female actually find each other on a deer. The deer is like the bar. The males don't take a blood meal. They don't need to. The females need the blood because they need the protein to lay eggs, but the male finds the female on the deer host. And so you've probably maybe encountered a male and they will just cruise around. If they get on you, they're not going to embed. They're just like, where's my lady at? And so they're going to be wandering across you. Deer are not good hosts for Lyme disease. We do not find Lyme disease in them. If so, it's super low levels. Why? So somebody had an ingenious idea. This was a PhD student, Patrick Pearson at UMass, with his advisor, Steve Rich, is take some deer blood and mix it with the bacteria that causes Lyme disease, and the deer blood kills it. So there's promising avenues of research understanding what's going on here and potential avenues for preventing infections in humans. There's some great research coming out of the Felicia Keesing and Richard Otzfeld labs on how can we how can we control tick populations at a neighborhood level? This is probably all important to you if you have a house with a yard. I know we've done some of these things. So there's tick control boxes. Folks have investigated spraying different fungicides and seeing how they may impact tick populations at a local level. And then there's a number of research studies. Again, this study is coming out of the same UMass Amherst group, looking at the efficacy of different repellents in keeping ticks off of people. So repellents don't kill ticks, but they keep them from crawling on you. In the medical realm, it's exciting. We have a Lyme disease vaccine in 3rd phase through Pfizer. And lo and behold, guess where one of the vaccine clinical trial labs is? University of Vermont. So they started a trial last fall, 2023. A number of people signed up. They are testing the efficacy of this, fingers crossed. And then interestingly, the University of Massachusetts Medical School has developed a human monoclonal antibody to be used as a pre-exposure prophylaxis for Lyme disease. So this is something that may become available in humans in the future, where you get this treatment and it will provide seasonal protection against Lyme disease. I don't know how that's going to work in Vermont because we don't have seasonal transmission of Lyme disease. We have year round. So again, but this is promising and that we are moving forward. For tick-borne diseases, the message from the CDC as well as state of Vermont Department of Health is prevention is key. And you've probably all heard this. I don't need to beat it into you, but basically to protect yourself against tick exposure. So avoid areas where ticks live. That's number one, recommended from Department of Health here in Vermont. Wear protective clothing and gear, use insect repellent, and protect and check your pets. They show this picture, this diagram at the bottom where ticks can be found. You can find ticks in brushy areas, in wooded areas, and on pets and wildlife. So basically that's everywhere in Vermont. So where does that leave us as Vermonters? It's definitely something that has been a pressing issue and has changed definitely the lifestyle of our family members because we spend a lot of time in the outdoors. So here's Hannah. She went from 3 to 12, and here she is in a permethrin treated suit and gaiters. So if she's tick collecting with me or going into tick habitat, this is how she dresses herself. And then this is my other daughter, Lena. And so this day she was playing in the yard on the grass, playing Frisbee, and the Frisbee went into tick habitat. That is behind her. Do you see that? And if I see the kids in tick habitat, I am screaming out the window, ticks, get out, right? But Lena knew that the Frisbee went in tick habitat. She ran in, got her tick suit and her gaiters on, went into the brushy area, got the Frisbee, came out, chucked the clothes off, and kept playing. So we're definitely all going to need to adapt given what we know and what we're learning on tick-borne pathogens in Vermont. So we'll end it there and open it up for questions.
There were ticks here long ago, but they just weren't harmful.
The 15 species?
Yeah.
Yes, good question. If I go back to the list, let's see if I can scroll super quickly. This is super interesting. And in all our time out to collecting, we've only seen one other species. The majority of ticks that you'll find in Vermont are American dog ticks and deer ticks. The other ticks are so host specific that you'll really only find them on those hosts. So there's a bat tick, there's a winter tick. You've probably heard about winter ticks and their implications in moose survival in the winter here in Vermont. They're specific to cervids, so deer and moose. There's bird ticks, there's rabbit ticks. You're not going to find those anywhere but on a rabbit or in a rabbit burrow. We have found 2 woodchuck ticks over the course of 2.5 years of sampling. So I didn't mention at the beginning, but this project is we just started this project 2 years ago. So, but there's a fisher tick. You're just going to find it on fisher. So they're really specific to their host species. Whereas the brown dog tick, the black leg tick, the Lone Star tick, and the American dog tick are ones that you'll find crawling on our animals as well as humans. Yeah. But black leg ticks are generalists. So you'll find them on anything that moves.
And it dogs in college, I can be.
Good question. Yeah. Well, fortunately, we have tick prevention methods for our dog. I don't know what we would do if we didn't. Yeah.
Would be tick surveys and some of them. Guards, it's a little odd. 7 ticks for all.
That's a good question. So I could go to that map. So every year the state samples a different, they don't sample the same towns every year. I don't know if they choose them randomly. Sorry for the flash forward here. So these are the towns that they sampled in 2023. If you look at the data from 2022, it's a different assemblage of towns that they sample. They'll send a crew out to collect ticks, and in Chittenden County, it was 3 towns. I don't know if they did those 3 towns on the same day, but they have a set transect length that they run. And so they may just go during the summer when the ticks aren't that active or there are not that many there. But the idea here is that they get broad representation across the state versus broad representation or, you know, focused on tick sampling within one geographic area. Yeah.
It's not like a minimum number of ticks per area.
It is, and it's based on a set length of a transect length. I think I'm not sure what the length is for the state. It might be like 200 meters or 250 meters total. So they just go to a site, measure 250 meters, pack up, go to another site, measure they sweep their drag cloth for 250 meters. So that's their standard protocol. It might be 750 meters, but I'm not sure, but that's their protocol. Yes.
I have 2 questions about the plasmosis and the Fuchsia.
Yes.
So in the animals that they've been found in, are they as bad? They have bad symptoms as humans can get from it? And
Good question.
have like the stages like Lyme disease where you treat it forever, you know, like
It doesn't, so anaplasmosis and babesiosis, I mean, I'm talking outside of my expertise. I'm a biologist, but it seems that they don't cause the same long-term issues that an infection with Lyme disease does. But in dogs and horses, they do have severe infections with anaplasmosis. So I know personally people who have had dogs with anaplasmosis, they just become lethargic and they're tired all the time and they're not eating and they're just fatigued. And so they can get over it with bacteria, with antibiotic. I'm not sure if they get over it naturally. And in horses, it's pretty serious. Good question. Yeah, but it is a pressing issue and a growing issue for domestic animals.
so like the mice that have that deer. I think that's so fascinating. I didn't know that before. Your blood feels hard.
It's so cool. I mean, we've known for so long that deer don't get infections, but nobody actually did that simple experiment. Just meant taking blood from a deer. They took it from uninfected deer and just in a petri dish. Yeah, I mean, we could all have done that, right? But this made the news. It's a really prominent finding discovery. Yes.
2-part question. I'm told that if you're bitten by a tick, you have like 24 hours to remove actual transmission
Yes.
to you. Is that accurate? Number one. And then the second question is the medical community treats tick bite with a single dose of doxycycline.
Yeah.
You happen to know the effectiveness of that?
That's a good question. I'm not in for the medical stuff, I'm not so sure. So I do know for the time of attachment and how long has that tick been embedded and like united with your bloodstream. There are various studies mostly with mice models, where they'll take infected ticks and let them feed on uninfected mice. And a lot of one of the studies that a lot of people were using for a while, and it's been duplicated, but shows that within that first 24-hour period, the 8 mice that they tested, one of them developed Lyme disease within 24 hours, and then most of them it took 24 hours, 36 hours, or 46 hours of exposure. So I don't think that the data are black and white. I think it's more gray, fuzzy. There are some newly emerging pathogens in ticks including another Borrelia species, Borrelia miyamotoi, as well as people are probably familiar with Powassan virus. And we know that those can be transmitted within hours of a tick biting you. So I think there's still a lot more research that needs to be done. The prophylactic treatment, that is something that's been actively researched and debated. So I'm not sure what the consensus is right now, but I'm glad that we're moving forward and being proactive after exposure to a tick.
We have one question online. Have you found tick numbers to be more prevalent in particular habitats, dry versus wet, etc.? I seem to find a lot of them in dry sandy.
Good question. So yes, so ticks, it depends on the life history stage. So you saw those larvae, they hatch out of the eggs, and you can expect that they're prone to desiccation. They don't really have a hard exoskeleton. So they need nice, moist, shaded forest floor. The nymphs, again, are smaller and prone to desiccation. So they're host seeking in the shady forest floor. Whereas the adult ticks, those are the ones that climb up and they will quest or host seek at the top of vegetation because they're looking for their primary reproductive host is a deer. So you're not going to find a deer on the forest floor. You got to climb up and hope a deer walks by you. So ticks are prone to desiccation. They don't like hot, dry weather because they will dry out. So it really not so much a sense of habitat type as more of climate conditions, weather conditions. So you won't find them as often in open sunny areas without any canopy because again they're going to dry out. So it all depends on the life history stage. But they are, in Vermont, you can pretty much find black-legged ticks almost everywhere, especially if you're on the edge of a field or in the edge of a sandy area, you're going to find ticks. And then you also need, when I take my students out in the field to teach them how to tick collect, I have them think like a tick, but you also need to think like a small mammal. So these things are going to be congregated where the small mammals are because that's their host for these first life history stages. Where are the small mammals in the environment? So I will say, I've had people come up to me and say, I have this sunny, dry field and I have ticks in it. Well, then you must have a lot of small mammals in there. So the ticks are going to be where the small mammals are. I'm not a mammalogist. I'm learning more and more about mammals, mostly for this project. My background's in ornithology, but I would just say, if you have small mammals, you're likely to have black-legged ticks.
Time for 2 more questions.
Are there insects or other animals that feed much on ticks? I understand that possums actually eat ticks, but I don't know if that means that they can sense them and seek them out and find them, or if it's just that they are good at getting them off themselves.
They're amazing at pruning. I don't think that they're finding the ticks or the ticks are finding them and then they will prune them all off.
Are there insects like type of wasp, let's say, or
Good question.
predatory spiders?
Yeah. When I was talking about the stupidly complex system that there is, nobody really has been focusing on the predators of ticks in ecosystems. There's actually an undergraduate student in David Allen's lab at Middlebury. He also studies black-legged ticks in Vermont, who's looking at spiders and other predators and how often they may feed on ticks and how they may play into this all. So, yeah. But some animals are amazing groomers, like opossums. Deer, amazing groomers. Moose are not amazing groomers, and that's probably one of the reasons why they've become so infested with winter ticks here in Vermont. They can have between like 50,000 to 100,000 ticks. They're just not good at grooming. But some animals are fantastic, so they'll be reducing tick population density. So if you have opossums on your property, welcome them. They are they're amazing neighbors to have. Yeah.
What about DNA? Is there anything you could do potentially for DNA and try to replicate the types of things that they've done other tests?
So like genetic modification?
Yes.
So there are some studies that are in early phases of genetic modification, so of small mammal hosts. So again, you know, people always think about the ticks as the bad ones, but it really, and I hope this message came across in this talk, that is actually the infected small mammals in your communities that are sourcing the infection. So definitely methods to make them less prone to infection would help the situation. So I'm not too familiar with that research, but I know there's research going on in that.
Are chipmunks and squirrels?
So chipmunks are really good hosts, and they're also good reservoir hosts. I didn't talk about them today because we haven't screened any yet, but we are in the process of screening them. So we have a roadkill permit through the state of Vermont where we've been picking up roadkill chipmunks. My kids can attest that mommy pulls over all the time to pick up roadkill. Girls, do I pick up roadkill? So chipmunks, red squirrels, gray squirrels, and rabbits are things that are not provided to the Natural History Museum. So we have gotten a number of samples. We're picking them up. I mean, I have to say biologists are resourceful folks, right? We are very resourceful. So I will have further results on, because they're there too. I mean, there's rabbits, millions of chipmunks. And we know that they're really common around disturbed areas, around human habitations, houses. So yeah, that's something that we're looking into. We'll be doing that screening in the next few weeks. So if you check back in with me, I'll be able to tell you what we find in chipmunks in Vermont. We just didn't get as many, but and they're all plucked off of roads.
Michelle, do you have one last final question? Sorry, your hand was up.
Oh, yeah, I'm thinking like a small mammal. And I'm thinking like a small mammal in Vermont. Who have a long history with these ticks.
Yes.
I'm curious about how the population of small mammals are reacting to this tick.
So if there's any like pathology that we could see or a negative impact of infection on them, that's a question I have too. I don't know if anybody's looked into it. It'd be interesting. That would be something that would be ideal in like an experimental setting where you're able to follow these small mammals. You're able to experimentally infect them and then follow them, and then measure variables of health. I don't know if anybody's done it in the wild, but that's something that I've always wondered. For a number of projects in our lab, we collaborate with veterinary pathologists. So I've been thinking about some of these small mammals should go off and see, you know, do they have enlarged spleens? They have myocarditis or anything like that, because they haven't evolved with these pathogens. They're novel, not just to humans, but also the diversity of other wildlife there. So really good question.
Great. Well, thank you so much, Dr.
Yeah, same.
Fantastic.
Yeah. Thank you folks for following.
Yeah, thanks everyone for joining us for the talk this month. And later this month on Saturday, September 28th in the afternoon, we will have a walk led by field naturalist alumnus Michelle Giles right here on a future vision on Centennial Woods at the UVM Centennial Woods Natural Area. After this talk, I'm sure all of us will be tick cautious while walking around at Centennial. So really appreciate that. Next month's talk will be on October 15th at 5:30 p.m. at Echo. Dr. Weston Testo will be presenting to us about the wetlands of the Champlain Valley. So hope to see you there. Thanks, everyone. And on your way out, please feel free to help yourself to cheese, crackers, veggies.
Lena, were you helping yourself to all those things? It's an accident.
Thank you so much.
Yeah, thank you for having me. It was fun. Mom, is that recording at the top? You're talking. You hear what? You hear you were hearing like a mouse across the front of the coffin. I didn't kill
Thank
this
you.
one.
I just wanted to say this was very interesting. Yeah, appreciate it.